Good afternoon. I'm happy to introduce Eric Barnett. He's in his third year of the MD PhD program in the Department of Neuroscience. He's been working with me on developing a deep learning, machine learning algorithms to predict ADHD using gene sets and polygenic risk scores. So Eric, I'll turn it over to you.
Thanks. So today, kind of the main meat of the talk is going to be predicting ADHD using gene set polygenic risk scores. But I'm going to start by talking about more simple models, and then I'll end talking about more complex models that I'll be doing in the future. But before I get into that, and because it didn't fit anywhere else in the talk and I didn't really want to break the flow later, I wanted to first talk about overfitting and how we combat that in our work. So if you're not familiar with machine learning, you should know that overfitting is a massive problem in pretty much all complex models. And it's because as you get more and more parameters that you can teach your model, it becomes easier and easier for that model to memorize the training data that it's trying to learn on. So here we have an example of underfitting, an appropriately fitting model, and overfitting, right? So underfitting, it's not really modeling where it should be correctly. It doesn't have enough parameters to bend this line. And finally here you see that it's separating the X's and the O's, but it's missing some, but you would expect it to miss those since it's interspersed in the O's. But here's an overfitting model where it's wrapping all around. And if you had a new distribution of X's and O's that came in, you would expect some O's to be in here, right? So this model, if you looked at the training accuracy, it would be 100%, right? And you'd be like, this is a great model. But if you looked at some withheld test set, you would get much lower performance because there are probably some O's in this area that your overfit model has memorized to call X's. So in machine learning, there's a bunch of different ways that we combat this overfitting. But in presenting results, one of the best ways to make sure that you're actually generalizing to a set outside of what you've trained on is by subsetting your data. And this means all of your model is trained on some portion of the data. It's the largest portion generally. And here I split it 70% of the data into a training set. And then there's a validation subset, which you use to optimize the hyperparameters of your model, which is essentially just all the options, right? You have all these options for your models, like the number of layers, how fast your model learns, the number of nodes in a layer, which is, I may get into later on in the talk what that means. And then you have your test subset. And this is when you test how well your model is performing in a completely withheld set. So the model wasn't optimized towards this subset at all. So it gives you a better representation of how well your model is actually performing. And all of these concepts will come back later in the talk. But I wanted to start with just some background on the genetics of ADHD. So for ADHD, the genetics account for 70 to 80% of the risk. This has been determined by twin studies. It's very hard. Oh, I probably paused there. The heterogeneity of the risk makes it hard to track down. This means that some group of people will have a bunch of mutations that produce a phenotype, whereas a different group will have an entirely different set. So many different mutations produce this same ADHD phenotype. And that phenotype is likely to just be a representation of an extreme of one or more heritable quantitative traits. And this is just a trait that is kind of on a continuous distribution. You can think of height as a heritable quantitative trait, right? It's has many different alleles across your genome that contribute to it, and as a result, you have differing heights. So you can think of maybe attentiveness as one of those heritable traits that we can't quite measure just yet, but ADHD is likely an extreme of that. And it's also been found that there's genetic overlap with ADHD and just many other disorders like schizophrenia, bipolar disorder, major depressive disorder, autism spectrum disorder, and many, many others. And finally, in 2019, there was a massive study. You can see there were over 55,000 total people in the study, and they found 12 independent loci that were associated with ADHD at the genome-wide significant level, which is a p-value of 5 × 10⁻⁸, which is ridiculously small. But even those loci that met that stringent criteria had very small odds ratios. So individually, they're not that important, and they account for a tiny fraction of the risk. And I put that in quotation marks because the authors of the paper don't even specify how much of the risk it accounts for because it's so small. And to get around this, what actually happens is you use all of the SNP risk across the genome, even if it doesn't meet the genome-wide significant p-value threshold. And if you combine all of that risk, it explains a much larger portion of the risk. In the 2019 paper, it was 22%. So that leads to the usage of this polygenic risk score. And what that is you have your genome-wide association study, GWAS, and it looks at the prevalence of each mutation or SNP in both the cases and controls, and it compares them. And it uses that comparison to determine a risk at each SNP. And as I said before, individually, those SNPs are not very useful, but together they can be much more useful. So what the polygenic risk score does is it sums all of that SNP risk across the genome. And generally, this is done by using different p-value thresholds. But in practice, the best p-value threshold is generally 0.05, but it's not much different from 0.5 or even 1. So you could use all of the SNP risk and you don't really care about all of the false positives, mainly probably because it balances out between randomly thinking that something is associated with a case versus randomly thinking something is associated with controls, and it balances out. But it's very interesting. So that brings us to the first question, which is how useful is this polygenic risk score alone in predicting ADHD? And we see here, this is the polygenic risk score on the x-axis, and the y-axis is just the number of people that had that polygenic risk score. And you can see that for ADHD, there's this much larger portion with a higher polygenic risk score, and that allows the model to separate out those cases, and it gets fairly good prediction. This is the AUC in the test subset, and I have a few slides on just what exactly AUC is, and it's the area under the receiving operating characteristic curve, which is normally abbreviated AUC, sometimes AUROC. But it essentially represents the degree of separability between groups in a model. And how it's constructed is you take each threshold in your model and you calculate the true positive rate and the false positive rate at each threshold. So you calculate it here, that plots to here, and then you keep calculating it all the way throughout your model. And the perfect AUC would be 1, which is represented here. Then you have a good AUC, which here is 0.7. I think this actually looks more like a 0.8 AUC, but that's okay. But you can see here that it's not completely separated now, right? But you still get good separation. You can get good predictability from this. And then you have your null AUC, which is 0.5, where you're getting no separation between the two classes here. So if we go back and look at the polygenic risk score, you can see it's kind of, I'd say, between what we saw for the good AUC and the null AUC. Luckily, it has this bimodal distribution that kind of saves it, because if you looked at this, you wouldn't be able to get much separation at all. But you can see the ADHD cases are much more likely to have this second peak here, which gives us that 0.64 test AUC. But now remember that on the background for genetics of ADHD slide, I said that there is some genetic overlap, which is the shared risk between ADHD and some other disorders. And what we did next was we used these genetically overlapping disorders to try to better predict ADHD. So our questions were, can these risk scores from those disorders predict ADHD? And also, is that risk different or complementary to the risk explained by the ADHD polygenic risk score? Because it's the idea that if something is predictive, but it's predicting in the exact same way, it's not actually useful to combine them together. But we ran these analyses, and we found that they did help a little bit in predicting ADHD. So remember in the last slide, we had the 0.64 AUC with just the ADHD training subset. And then here we looked at individual polygenic risk scores from those other disorders. And we found that some of them were predictive by themselves, right? So alcohol dependence, bipolar disorder was very predictive, almost as predictive as the ADHD training subset, which is interesting. Major depressive disorder was predictive. And then combined together, they did pretty good. You could see that it's largely driven by bipolar disorder, since it's barely improved. But overall, if we combine all of these overlapping disorders with the ADHD training subset, we get a 0.02 improvement in the AUC, which might not seem very important, but generally in machine learning, improvements happen in small jumps. You don't all of a sudden jump up 0.2 AUC. If that was the case, all machine learning would already be perfect. And how we improve machine learning is incrementally making all of these little improvements and eventually getting to something that's more useful. So with that in mind, we can see that you can see all of these density plots that aren't really as impressive as the ADHD density plot I showed earlier, right? There's not a whole lot of separation between them, right? But together, they were able to better predict ADHD. And with that in mind, we wanted to know if instead of looking at all the risk summed across the genome, if we could break that risk into smaller subgroups to better explain risk and better predict ADHD, which is where gene set polygenic risk scores come in. So the gene set polygenic risk score are a sum of the SNP risk across a gene set for each person, where before it was the sum of the risk across the genome. And the questions we had implementing this new method was, can combining multiple gene set polygenic risk score better model the risk and better predict ADHD? And the rationale behind why I think that might be the case is this strategy is more resistant to the heterogeneity compared to just looking at individual SNPs, because the idea is that you would be combining similar SNPs. So maybe one person has SNP B, another person has SNP A, but they have the same kind of risk. They're affecting the same biological process. And if we group that together, we can get around that heterogeneity. But this gene set polygenic risk score also has more potential for learning patterns compared to the genome-wide polygenic risk score, right? When you look at genome-wide, you just have one value. It's really hard to find patterns when you're looking at one value. Generally, you just have to set a threshold, and everyone with more risk than that threshold will call a case, and everyone to the left of that threshold, I guess, is a control. When you have more risk scores, you can subset into smaller groups and hopefully better predict the disorder. So here I have the density plots for specific gene sets. And here it's even more apparent than in the other disorders density plots that there's not a whole lot of separation in these density plots, right? But then at the end of the day, we combined 50 of them and also add in the polygenic risk scores. And you see a pretty massive separation, relatively massive to the other separations that I've shown you so far. So here we can see that adding up all of these different risk scores better allows the model to learn the patterns that are in the genetics and learn how to separate ADHD cases from controls. And this plays out in the AUC values too. Remember from the last slide, we had the 0.66 AUC. And now if we take everything that was in this model, but also add the top 50 gene set polygenic risk scores, we jump up 0.06 AUC, and now we're getting something that is more reasonably performing at 0.72 AUC, which plays out in this separation. But there was also a question of whether we can use these gene set polygenic risk scores to separate ADHD cases and controls just by themselves. So ignoring the genome-wide polygenic risk score, can we still successfully predict ADHD? And so here I had a bunch of different models that had a different number of gene set polygenic risk scores. And they were by themselves pretty predictive of ADHD. And you can see it quickly plateaus, like 25 gene sets was non-significantly better than 50, which means that it probably reaches some plateau, and adding more gene sets after that might have not helped us. But even at 5 gene sets, we get 0.64, which you might remember was the original AUC that we had for just the ADHD training set. So we were able to replicate the performance of the polygenic risk score by just using 5 gene set polygenic risk score. And I'll show on a slide later, but this is actually using 6 times less SNPs after all the pruning that goes into it to get rid of correlated SNPs, which is pretty interesting. So if you're paying attention and hearing me talk about top gene sets, you might be wondering how exactly we determined the top gene sets in the model, because at the start we have over 10,000 gene sets. So how do we get down to 50 from there? And this slide definitely could use some optimization, and I've highlighted that with the blue stars. So the blue stars represent a number that could be optimized, and if I spent all the computational power to optimize all of these, we'll call these hyperparameters, I guess. If I optimized all of them, I think I could get probably slightly better results, because there must be a value that performs the best here. But these are just the values that I selected. There is a little bit of optimization, but not much. So we go from 10,000 gene sets down to the 200 gene sets that a MAGMA gene set analysis found were most associated with ADHD. And how it does this is it looks at the SNPs within the gene set and kind of sums them up, similar to the gene set polygenic risk score, and looks at how those gene sets are collectively associated with a disorder. So we selected those 200, and then we created the gene set polygenic risk scores using the SNP risk found in not only the ADHD training subset, but also from the summary statistics of these other related disorders. So from that, we used ADHD plus 5 other disorders. So we multiplied the 200 top gene sets by 6, and now we have 1,200 gene set polygenic risk scores. But I don't want to throw all 1,200 into the random forest model, which I'll explain in a little bit what that is, because that probably wouldn't perform well. So what I wanted to do was select out of those 1,200, 50 that would perform best. So how I did this was I started by randomly selecting 100 gene sets out of that 1,200, and I trained a random forest model and calculated gene set importance. So now how random forest models work is they're a collection of these decision trees, and for each decision tree, a random subset of the data is thrown into a decision tree, and then it makes these rules that try to split the data into smaller and smaller groups. And the hope is that at the end, you have a subset that is either entirely cases or entirely controls. So it takes these random subsets and puts them into a bunch of trees. So then you have a random forest. So with that random forest, I calculated the importance, and the importance is essentially how often that feature is used in rules, right? Because if it doesn't split the data into nice groups, it's not very important. So after that random forest, I kept the 50 most important gene sets, and then I randomly added 50 more gene sets in, and trained another random forest model and calculated the gene set importance again. And so then I repeated this step 5 500 times, just to make sure that every single gene set had multiple opportunities to kind of ingrain itself into the random forest and become important. If these models run pretty fast, if the model was going to run slower, I probably would have done less iterations than this. But that's how we determined the gene sets that we used, and what those gene sets were is presented here. And I'm still going to, in a couple weeks, I'm actually running this analysis on an even larger data set that's about twice as large as the data set I used here, which was 10,000. So as I said before, at the lowest number of gene sets that we used, the 5 gene sets, it had a total of 52,000 SNPs that were used. And it had the same performance as the genome-wide that used 322,000 SNPs, which is pretty interesting that we're able to get the same results using 6 times less SNPs. And what I noticed looking at these top gene sets, so here I have their rank of importance based on the random forest feature importance. And you'll notice that it's not actually linked up to the magma association rank. So the gene sets that were most associated with ADHD aren't necessarily the top most important features found by random forest. You can see that 2 and 3 are here, but there's a bunch of less associated gene sets. And why I think this might be the case is that it might actually be sharing risk information. So random forest doesn't need to repeat risk rules. If a risk rule would create the same subsets, it's not important. It would toss that out. So that might be why we're seeing that difference between random forest feature importance and magma association. And I also wanted to note that these results are pretty largely driven by the gene sets found in the ADHD subset that we used, but not entirely. There were 2 major depressive disorder gene set polygenic risk score, 2 alcohol dependence, and 1 anxiety in the top 50 gene sets, which I think is pretty interesting because it kind of says that the SNP risk from those disorders actually better represents the risk in ADHD than even the ADHD SNP risk does, which is very interesting. And so this is where the results that I'm presenting today ends, but I have some next steps for the project that I wanted to share. And the first one is a direct extension of this gene set polygenic risk score idea. And the idea behind this is creating more predictive gene sets. So the skeptics out there thinking about the gene ontology derived gene sets are definitely thinking that these gene sets are incomplete and have errors in them. Everyone that I talk to about gene ontology has this same understanding that there are errors among them. And there are also differing evidence bases for each gene set. So maybe some gene sets are actually very good, but others could be not so great. And probably most importantly, they're not optimized for ADHD prediction, right? The rationale for us using them was to try to capture SNPs into the same molecular function or biological process and hope that it's a similar risk, and so we can kind of get around the heterogeneity. But what might be better is if we allow machine learning to create its own gene sets. So which collections of genes would best allow the model to predict ADHD? And we have the tools to do this with neural networks, which take some input. Here it would be if we collect the SNP risk within a gene, and then we have a sparse neural network, which means that each input isn't connecting to every node in the next layer. And what this is, each line represents a transformation of that input. So let's say gene 1 risk, you multiply it by 2 and then you add 5. And the multiply by 2 and add 5 are trained in the learning process of a neural network. So here you can have a model that creates its own gene sets that would hopefully improve prediction. And these, I think, have even more potential for learning patterns than the gene set polygenic risk scores, because you don't have all of this potential extra SNPs in each gene set. You only have the SNPs that the model determined were important in that gene set. And one of the things to keep in mind here is this is definitely an area where you would need to be careful to not overfit, because there are a lot more genes than there are gene sets. So if you use all genes as input, you have the potential to have a lot more parameters, which, as I explained on the first slide, gives you more potential to overfit. But that's definitely something that we're going to keep in mind. So now I wanted to recap everything that I've said so far to hopefully lead us in the next direction. So I started by looking at the polygenic risk score only and using it to predict ADHD, and that established a baseline performance that was it was pretty good. So then we used that polygenic risk score, and we used the polygenic risk score derived from the overlapping disorders, and we saw a small improvement in the prediction of ADHD. And then from there, we went to using all of those polygenic risk scores, the genome-wide, and adding in these gene set polygenic risk score, and we saw even more improvement. And then I just told you that now I'm interested in looking at the risk compiled in genes and seeing if we can use those the SNP within genes to create better gene set polygenic risk scores, and maybe see even more improvement from that, hopefully. So then, you know, I'm going from genome-wide to gene sets to genes, and the question might be, why don't we just use individual SNP risk as input into our models and avoid grouping them entirely? We can get down to the base immediately. But this isn't done for a few reasons, but one of the biggest reasons is that if we used every SNP, there would be far too many parameters to train, and the model would not converge, and it would give you very poor results. So if you think about the number of SNPs in the study, and in the data set that I'm currently using, there's something like 1.8 million SNPs. So let's say your input layer was each of those SNPs. So you have 1.8 million lying down here, and then you're connecting to further layers. So you would need 1.8 million times the number of layers you have in your nodes you have in your second layer. And you can see you're rapidly getting a massive number of parameters. You want to train each of those parameters, but how would you train so many parameters at the same time? You eventually could if you had enough examples in your training set, but you would need millions of people with ADHD and millions of controls to get there, and it wouldn't be worth it. And if you decided to trim down that list, if you maybe use the most associated SNPs, you would be missing way too much information. Remember in the first slide, the genome-wide significant SNPs explained very, very little of the, they had to use all of the SNPs to explain the risk, which kind of gets us to this point where how do you decide how many SNPs to use, and it becomes a big problem. But we think we have a method to hopefully solve these issues. And that method is using convolutional neural networks to look at the risk. And I have this slide that I like that explains convolutional neural nets. But they come from the image recognition field, which is one of probably the top machine learning field, has the most advancements in it. And here you have these things called feature detectors. And the feature detectors scroll across an image and try to look to see whether that feature is there. And this is a bit of a simplification because I've put these weights in by myself that to me kind of look like a 2. In reality, the model trains these feature detectors by themselves so that the model can decide what features it thinks is important, not you. But this is just an example. So it scrolls across the page and it's like, is there a 2? No, 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 there's finally a 2. So it says, yes, there's a 2 there. So that's a feature detector. And then we have another concept for convolutional neural nets, which is called max pooling. And here I'm asking the question, was there a 2 on the slide? So I look at this, this new feature that's been generated, right? So it looked at all the pixels on this last slide and it condensed them down to, what is this, 9 features. So there were a ton of pixels here, and now it's just represented as 9 input. And then I can pool the those input across some range, and I can look for the max value. And here it would really be a number, but here I've represented it as no's and yes. So here the max value is yes. And you can kind of begin to see how using these methods, we can drastically reduce dimensions from an initial layer that has all of the SNP information to further down layers that have a much more condensed representation of the risk. Which leads to this idea of a genomic convolutional neural network. And here we plan to detect and pool the risk across each person's annotated genome. And as I said before, this reduces the parameters necessary as it condenses the information, which allows us to use more SNPs as input. And you'll have to excuse my terrible drawings. It looks like I've actually drawn them with my mouse, but I haven't. I drew them with a pen. But here we have the risk on the y-axis and the genomic location on the x-axis. And we can pretend that this is a person, this is a different person, and this is a third person. And we want to know the risk across the genome for those people. So here we would have a bunch of feature detectors. And like I said before, here we don't know what's in the feature detector. The model trains it itself. But the idea is that we have all these feature detectors and they're scrolling across the genome and outputting some risk at each larger location. And we can use that risk in further layers of the neural network and hopefully try to better predict ADHD or whatever disorder that we're using it on using this idea. And I mentioned that it's an annotated genome, and this is kind of just looking at the risk. But we also want to look at each location, give the model a better understanding of what's actually at that location. So in the data matrix that goes into this, we have all these annotations at each location to try to give the model the most information so it can try to find those patterns similar to the gene set polygenic risk scores. And at the end of the day, hopefully improve our ability to predict these disorders. So that's all I have. I'd like to thank everyone in the Faraone lab, in the PsycGene lab, and my advisory committee. And I'd be happy to take any questions. Oh, and now I see that there were questions in the chat that I missed. Yes, and Cindy asked why I didn't use the schizophrenia polygenic risk score in the analysis. And it had something to do with who we were producing the first analysis for, but we definitely will be using schizophrenia and autism that both have large overlaps with ADHD in our future work. So that's, I'm definitely looking more into which polygenic risk scores might better improve. But yes, the schizophrenia overlap is smaller, but it'd be interesting to look into which overlaps might be important. I mentioned that some of them might be complementary and offer something that isn't found in the disorders polygenic risk score that you're looking at. So like the ADHD polygenic risk score.
Eric, could you talk a bit about incorporating ancestry into the model and why that's important?
Right. So I avoided it here to avoid any complications that might come across there. But one of the other large issues with machine learning models and just modeling the risk of these disorders on the genetic side in general is population stratification. And that's this idea that instead of predicting these SNPs that are actually causing risk for the disorder, what you're doing is predicting ancestry. And you're finding these groups of subtle ancestry that just happen by chance to have more cases than controls in your sample. So you could, maybe there's one small ancestry that by chance has 100% cases in this sample. In reality, it's not the case, but just by chance. And so you would learn to use the ancestral SNPs to predict the disorder, but we definitely don't want that. Those are false positives for sure. And we're combating that in our more complex model by doing this adversarial task. So we're telling the model not to be able to predict ancestry from all of the layers in the model. So when it trains, it'll see if it's able to predict ancestry. And if it is able, it thinks that's an error and it tries to get the model back to 0.5, not being able to predict ancestry at all. And that's one way that we're combating the ancestry problem in population stratification.
Hey, Eric, this is Inshilin here.
Hi.
Nice talk. Clearly, it's outside of my area. So I've got a impression that it seems like the prediction power, like whatever the ACU,
Yes.
That you are talking about, the more information, seems like the more information you use, you get better prediction. That's sort of predictive, right? You started out with genome-wide, and then you started out with certain genes, and then you started gene sets.
Right.
And it's sort of consistent with that. If you use more information, then you get better prediction. Do you think it's a way that you can find the key piece of information that will give you the predictive power without using all the other information?
Right. So that kind of is the case. And in the gene set polygenic risk score, it's not like we're using new information. All of those SNPs are already contained in the genome-wide polygenic risk score. we're just splitting them into smaller groups so that hopefully we can learn better from those smaller groups. And there is this idea that using it, more information would allow your model to predict better. And that kind of makes sense to everyone, but you also need to be careful to not throw too much input into a model, because let's say we don't know exactly which input will be helpful. If we put a ton of useless input into a model that maybe we think might be helpful, it can actually confuse the model and make it harder for it to search through, the pile of useless information and find the useful bits. So it's definitely a balance that you have to strike between throwing all the information that you have in a model and not giving the model enough information to learn.
Yeah, that's very interesting. In that case, probably you can have a control to show the power of your model, to sort of feed your model with sort of unrelated gene sets and see how that affects your prediction.
Yes, that's a very good idea. And in practice, we almost have those controls because we have many features that aren't useful. But you're right, we could make useless information that we know is useless and see how it affects model prediction.
Yeah, thank you.
Thanks. And there are a couple more questions in the chat. So I'll just read those. Cindy says getting away from gene ontology seems like a good idea. As another concern is the unequal number of genes in gene ontology and the overlap within gene ontologies. And I agree with that. But then a question, are you only able to model additive SNPs? Oh, and I'm glad you raised that question. As could there also be protective SNPs or SNPs that counteract each other within a pathway? And this is a great question because the genome-wide polygenic risk scores only take into account the additive effect of the genes. But when we break it down into smaller portions, either the gene sets, the data-driven gene sets, or even at the SNP level at the end, we can input that risk into a model separately. And once it's inside the model, you can have those other interactions between SNPs, between genes, and between gene sets. So it could better model the non-additive effects of the SNPs. And it could also definitely discover the protective SNPs, right? So you could have a node in your neural network that is modeling protective features. If that helps the model better predict ADHD versus control, it will hypothetically turn that into a node or multiple nodes so it can learn better. And then Ryan asks, are there any known protective SNPs for ADHD? If so, are they used in the algorithms? And two, what are the implications of the fact that some other disorders' polygenic risk score can predict ADHD better than ADHD itself? And if we go back to that, it's not actually the case. So ADHD itself was at 0.64. The highest individual disorder was 0.61. But I think what you've mentioned there is actually possible. And my thoughts behind that are, if you have a training subset for finding this risk that's very small compared to, so the bipolar disorder study was a massive study. It was something like 50,000 subjects in it. So in that study, they're probably getting the best representation of the risk. And for the part of the risk that's overlapping, it's probably doing a better job at presenting that risk than disorders that don't have that many subjects. So it's not the case for ADHD, but I could definitely see other disorders being better just because they have more subjects that have been studied and it's representing the overlapped risk. So if you have enough overlapping risk and the overlapped risk is much better represented through a different study, it would be possible. And I think I covered the protective SNPs for ADHD. I don't, someone can correct me if I'm wrong, but I don't think there are currently any actual SNPs that are known to be protective, but it's definitely out there as something that's likely to be happening. And it's probably likely like the risk SNPs in that the effects are very small. So you'd have to combine them into, I don't know, maybe a resilience score. And then Cindy asked, is there a way to use transcriptomics to understand how combinations of SNPs within a gene could impact the overall gene expression? So inform the combining of the SNPs based on one of the likely steps the genetic changes influence. This is an interesting idea, and I think it's probably in the future of how machine learning models will actually be used to predict, maybe even clinically, like long downstream. Because here we're just looking at genetics, but in reality, the model that would best predict ADHD would be a model that uses gene expression and genetics and clinical features all together to look for interactions between all of those different omics and all the different clinical data that you can gather to create, the model that has the most useful information and can be most informed when it makes its predictions.